Here I'm going to go over a gene editing technique gaining a lot of popularity in the scientific community called CRISPR. So CRISPR, the basics of it is that it's a gene editing method that exists in living organisms. The organisms that it exists in it's a simplified antiviral defense system found in bacteria. Scientists have been able to identify this and are now using this for gene editing of many different cells other than just bacteria. CRISPR allows for cells' genome to be cut at predetermined locations. It's very specific, which is part of the reason why it's getting popularity, and that will result in the removal or the addition of specific genes. So it's specific and it allows for modification of those genes by either addition or removal. Now, CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. So I tried to highlight the words here. Clustered is just kind of a grouping. Regularly, it's a defined pattern. Interspace is just a distance between objects. It's small and short distances, so it's looking for a very short sequence. Palindromic, that's a sequence that reads the same backwards as it does forwards, such as mom or dad, or something a little more complex like race car. If you spell the letters out forwards and backwards, you'll get this same word. Remember though, DNA will always read in the 5' to 3' direction. And the last letter R stands for repeats, and this occurs again in the same form, kind of like the post here. So a lot of times you'll hear CRISPR referred to as CRISPR Cas9. The reason why that is that Cas9 is an enzyme that can cut DNA. So CRISPR Cas9, the complex, allows for the removal or addition of specific genes, which is great. The possibility via Cas9 nuclease, remember those 3 letters, ACE, can imply an enzyme. In this case it does. This is a complex that's created with guide RNA. So the Cas9 in this image is kind of like the blue areas that you see, and the guide RNA is kind of the green areas here, and you'll see that through some of the other images in this presentation. So this guide RNA, this gRNA, guide RNA is sequences that will bind to the DNA sequence to be edited. It's often abbreviated guide RNA as G, little g RNA, or single guide RNA, sgRNA. This guide RNA is non-coding. It's a short RNA sequence that will bind to the complementary DNA sequence. So we see the double helix, double strand of DNA here, and here it's binding 5' to 3'. This sequence, which is the reason CRISPR can be so specific, it's looking for these specific nucleotides. That guide RNA is encased and wrapped here in the Cas9 enzyme. Now, the basic process of CRISPR is first, scientists have to identify target DNA sequences. So in the genome, that could be a gene that needs modification, that could be a disease, whatever gene is being identified first by scientists. Then it has to be identified so that guide RNA can be created so that the complement of the DNA sequence. It's very specific, it's a very short region, so we need to identify the gene and then make the complement RNA for the guide RNA. Then the guide RNA and the Cas9 are attached to form this complex, and then this complex will then be inserted into target cells. As it goes through target cells, it will go through and read the DNA. In this case, we're reading that chromosome, and once a sequence is found, the DNA will be cleaved. I have a picture of a cleaver here, or a knife. It's kind of cutting. It's going to cut that DNA. Once it cuts that DNA, that can allow for the addition or deletion of nucleotides to occur at that specific region. Now, the advantage of CRISPR, some of the possibilities, it can silence genes. It can alter disease-causing genes into a healthy one. Here's just kind of an example here of some diseases that have been identified on different chromosomes. So once they're identified, if we get the sequence, CRISPR could possibly be used to alter the disease-causing gene into a healthy one, not only to the benefit of the individual, but their offspring and lineage. Can also be used to specifically insert a codon, could be a stop codon or for a particular amino acid into a sequence. It can also change or alter gene expression, silence them or upregulating them. Other than editing genes, CRISPR can also activate the transcription process. And remember, transcription is DNA to RNA. How that's done is through the deactivation of the Cas9 cleaving abilities, meaning that in this case, the Cas9 will not be able to break or cut in any way, so it's just able to go through and activate the transcription process. It can also repress or silence genes. Here we see it as a repressor blocking certain genes from being expressed. And the last one here, adding a fluorescent label to visually identify physical locations of DNA sequences. This could be important where we're identifying and then it's going to add a little label. This can allow scientists to know the physical location of certain genes so that they can aid in development of a 3D model of the genome and tracking the chromosome movement. So that's another reason why this degree of being able to specifically identify certain regions, here visually marking them, can be advantageous to scientists. In addition, we have to remember that when we're modifying genes, there's also the potential for social concerns. Should human genomes be altered in somatic cells or embryos is a controversial issue, no matter how it's done. And CRISPR is just kind of the latest technology to offer this. What if undetectable off-target effects occur that have not been studied comprehensively? What potential negative impacts could that have? Well, this could change the human genome through generation and after generation, even cause disastrous consequences. So think about if you modify all the genetics of a lineage or generation now, even if you don't do it in the future, those modifications will be passed on. So these cause worries and include related to mental health, social behaviors, designing human capabilities such as intelligence, appearance, personality, vision, eye color, a whole bunch of different things. So there's just social and ethical concerns with this technology because it is genetic modification. There's also been some research over in the UK that have reported some unwanted results of CRISPR. So while CRISPR is often used to generate small deletions, as I've shown, and that's what it shows here, scientists have seen unintended large deletions. When I say large, we're talking several thousand nucleotides long, in addition to complicated DNA arrangements of distant sequences being stitched together. These are all things not intended, but can occur, so must be mindful, especially when we're looking at human gene editing. This phenomenon was prevalent in all the three types of cells tested, including a kind of human growth cell in the laboratory. So again, something just warranting caution, and this is a relatively new technology and needs to continue to be studied so we don't have any of these unwanted edits. Now with CRISPR, one that's been used in a positive note is what's called precision-guided sterile insect technique, kind of a long term there. Researchers have developed a new way to control and suppress populations of insects, potentially including those that ravage agricultural crops and transmit deadly diseases. Insects can mass produce, they're very R-selected species, so by controlling their populations, that can be advantageous. And instead of spraying a pesticide that could kill all insects, this could be very targeted. The precision-guided sterile insect technique, the PGSIT, alters key genes that control insect sex determination and fertility. So how could this possibly be used to control insect populations? Well, when we take these precision-guided sterile insect technique, what will happen is we'll take eggs and we'll introduce into target populations where only adult sterile males will emerge. This results in a novel, environmentally friendly, and relatively low-cost method of controlling pest populations in the future. This is also very specific. And the screwworm fly was the first pest successfully eliminated from an area through sterile insect technique by the use of this integrated area-wide approach. So it's not looking at spraying some sort of chemical that may enter the environment. This is using CRISPR to specifically target this particular species of fly, create sterile males, and really reduce and hopefully potentially eliminate them from the population. Lastly, because this is such a new technology, so it's looking at some sources here and also some additional YouTube videos that you're welcome to take a look at if you're interested in learning more about CRISPR.